Hello class. Today we're going to be looking at chapter 8, section 2, which is mitosis. You can see a couple pictures here of kind of what we're going to be looking at with the cell and how it divides and how it replicates. So first we're going to look at why do cells do this. And basically it's pretty obvious. If a multicellular organism needs to grow, it's going to have to add more cells. A cell itself cannot get larger and larger and larger and larger and still continue its function. We studied that back in a couple chapters ago. So cells cannot keep growing indefinitely. They cannot just keep getting bigger and bigger and bigger. And because of that, then they will replace or they'll repair any damaged cells, or if you're growing, they will just make more of themselves to allow you to grow. If it is a single-celled organism, then it will replicate in order to kind of as an asexual reproduction. So it depends on which type of organism it is. And our main things then are going to be growth and replacement of cells. Asexual reproduction is going to be single-celled life forms, and this we generally call binary fission. In sexual reproduction, we're going to look at meiosis, which is a process of cell division, and we'll get to that in tomorrow's notes. So eukaryotic cell division is called mitosis, and mitosis just means growth division. It's division of cells producing two identical daughter cells. So we get an identical cell. It's going to be a little smaller because we're making it from this original cell, but it will eventually grow to its normal size, and then if it needs to, it will also divide and make a new daughter cell. Meiosis, on the other hand, is called reduction division, and in this one we actually produce 4 haploid daughter cells. We're going to get to meiosis in the next set of notes, and so you can look forward to kind of hearing about that a little bit more. Meiosis is used when we are making egg or sperm cells in the human. So we're going to look at mitosis today and the cell cycle or the life cycle of a cell. And you can see here that this is going to be a picture image for you based on the life cycle of the cell. In your notes packet, you have one that looks very similar to this. So if by chance you are using that notes packet, you have this picture. If you don't have the notes packet, you may want to go ahead and draw something in like this. So we have 2 major parts, interphase, which includes G1, S, and G2. And then we have cell division, which includes mitosis, and then cytokinesis. You will need to know all of these parts and what happens in these sections. So we'll start with interphase, and this is the stage that your cells are in most of the time, especially now because you've hit probably an age where you're not growing as much. And so your cells are mainly in interphase. This is where they spend majority of their life. And this is the normal job or function of the cell. There's 3 stages, G1, S, and G2, which we'll go through here in just a minute. Most of our cells over here in this picture of the onion are in this interphase stage where it doesn't even look like you can see chromosomes. They're just kind of there. Just the cell, you can see the cell, but you're not seeing anything that's happening inside the cell. So this is kind of another image of our interphase process with mitosis and cytokinesis being a tiny little section of the life cycle here. We have G1 to put in some things that happen at G1. The cell doubles in size. Enzymes and ribosomes and mitochondria double in number. So this is called the growth phase. This is growth 1 phase or cell growth. In the S phase, DNA will replicate, and this will be important for making a new cell. Many cells might not ever go to the S phase. They might stay in the G1 phase. They've grown, and then that's where they are. They don't really make any more of themselves. And then if they are a cell that will need to replicate or needs to repair itself, then it'll go to the S phase. After the S phase, we have the G2 phase, and in this one, it's another growth phase where we get some extra enzymes and things like that that's going to prepare it for mitosis, for the division. Okay, so make sure you're writing in what happens. G1 is the growth phase. S phase is a DNA replication. where DNA builds an exact copy of itself. We're going to get to this a little bit more in chapter 10, and we'll talk about how this happens. But what happens during replication as a general model here is the double-stranded the new molecule will contain one old strand and one new strand of DNA. The whole process we will get to in chapter 10. You can see a picture over here. We had our parent strands, which was the DNA that was in the original cell, and then they split and add a strand on each side, a complementary new strand, and each of those then DNA, one will stay in the original cell, one will go to the new cell. Okay, so now we're on to the stages of mitosis. This is on page 156 if you'd like to look at some pictures from your book. So with mitosis, we have four main parts: prophase, metaphase, anaphase, and telophase. You can draw in some examples for yourself in your notes packet if you like, and give a little example or a couple sentences about what happens in each phase as well. So step one is prophase. In this one, our chromosomes form from the chromatin. So we start to actually see the chromosomes. And you can see down here in this picture here from the microscope that you can start to see these like worm-like chromosomes that are developing. The nuclear membrane will disassemble. So we start to lose the nuclear membrane. You can see that represented here. The centrioles will move to poles. And that's what these are, little centrioles. They move to the ends, opposite ends of the cell. And the spindle fibers form, and that's what these little fibers are here. Those will help to pull the chromosomes to each side of the cell eventually. Mitosis step 2 is metaphase. And this one is pretty simple. This one's really usually the easiest to identify, I think. Metaphase, all the chromosomes line up across the center of the cell. And this is where, if we're going to look up a karyotype like we made yesterday in class, the karyotype is formed from the chromosomes that they can see here. They're just a little bit easier to see and identify. And this is called, when they line up across the center of the cell, it's called a metaphase plate. So you may want to draw one of these examples in there. Step 3 is anaphase, and in anaphase, these chromatids separate and move to opposite poles. And you can see that here. They're getting pulled apart to the opposite ends. This is a real picture of an onion frozen in time here in anaphase. Mitosis step 4 is telophase, and in telophase we start to actually separate the cell a little bit. We can see here that it's going to be the opposite of prophase, so we have spindle fibers that are disappearing. We're not going to see those anymore. The nuclear membrane starts to form again. You can see that with the dotted lines in the drawing there. Chromosomes go back into chromatin, so we start to not see the actual identifiable chromosomes anymore. And then 2 nuclei per one cell. So we have a nucleus here, we have a nucleus here. We start to see this little kind of pinching off between the cells. That's going to lead us into our next stage where they actually do break apart and make 2 whole new cells. So our last part, which is really kind of its own thing, mitosis has those 4 stages, and then to end mitosis, we go into the next phase, which is called cytokinesis. And cytokinesis is division of the actual cytoplasm. You can see in the pictures there the differences between animal and plant cell. The cell membrane will pinch in to form 2 separate cells, and so you get 2 identical daughter cells. In animals, this is called a cleavage furrow. So that word there you're going to see again. Make sure you mark that down. And the cleavage furrow is represented up here where you start to see that break apart or pinch apart. In plant cells, you get what's called a cell plate. And basically that's the cell wall that is being formed in between the 2 nuclei. So you can see that there, that those are the cell plate that's actually going to be the cell wall. And then that will pinch off the 2 or separate the 2 cells. Okay, here's another example of that. So we have the cleavage furrow in an animal cell, and then we end up with two daughter cells. And they're both smaller than the original cell because they've been divided. And so we have two smaller cells that will grow until they get to their normal size, and then they will either go through division as well, or they'll just hang out in interphase and go through their normal functions. In a plant cell, you can see here the cell wall is created, that cell plate between the two nuclei, and then we end up with two daughter cells. Okay, to kind of show you how this works, we're going to use this website with McGraw-Hill. They have a great example of how this works, and just kind of watch this animation here. Okay, so you can see here, these are the stages of mitosis as we just talked about. We have interphase, prophase, metaphase, anaphase, and telophase, which leads into cytokinesis, and you can see kind of that separation of the cell. This shows the same types of images here just for you to see: metaphase, anaphase, telophase, and cytokinesis. And this is what it looks like in an onion cell that has kind of been frozen in time. So you should be able to identify some of those stages here. And you may want to practice that. Just kind of look here and see if you can identify some of those stages. Okay, the last thing for today is asexual reproduction, and this is an exact copy of the parent. So this is going to be in like single-celled organisms, things like that, that go through asexual reproduction. Bacteria, molds, algae, and protozoa are example. And all of the cells form through mitosis in this situation because they don't have sexual reproduction, so they don't need meiosis to happen. And so some examples of asexual reproduction are regeneration, or they can regenerate maybe a tail or something like that, budding, spores, you can see a picture there, and then binary fission, which is basically like mitosis. They kind of separate into two separate cells. Okay, and that completes mitosis or cell division.